Hello, everyone. Today I'll be talking about TCRBinder. Just to give some introduction, um, there's some special components of the adaptive immune systems, which are called T cells, and they have a receptor called TCR. And this is really important because this receptor can recognize some peptides in virus or in cancer cells called epitopes. And these interactions help those T cells to actually do the killing, which is what we want. So a current challenge in computational immunology is to be able to identify whether there's going to be binding between a given pair of TCR and epitope. So here I'm just creating a convolutional neural network that predicts whether there's going to be binding or not. Just to give some more backgrounds, given T cell receptor contains an alpha chain and a beta chain, and then the beta chain contains a specific region called the CDR tree, which is considered as the primary determinant of specificity. And that's what the majority of the databases contain. So for this specific task, I'm using a database called PDJDB that looks like this. It contains a lot of experimentally validated pairs of CDR trees and epitopes. You can see the sequence here. And I processed this data before training my model. And what I did was to keep only TCR beta sequences coming from human, and I am only considering epitopes that are being recognized by more than 50 TCRs. Here you can see the top 25 epitopes and the number of TCRs that recognize those epitopes. As you can see, many TCRs can recognize the same epitope. I'm just showing this sequence logo for this epitope right here. And this logo is pretty much a consensus of all the TCRs that are able to recognize this antigen. I hope you can appreciate here the positions 6, 7, and also from 10 to 13. There's some conserved amino acid residues present in more than 50% of the TCRs that we're hoping that our model learn to be able to predict binding or not. This is just some distribution of the number of TCRs that recognize the epitopes before the filtering. And then in red, you can see the distribution after the filtering. One major problem for this task is the absence of negatively labeled data, meaning that there's, at least not to my knowledge, a database that contain pairs of TCR epitopes that do not interact. So what I did here is to use my database and then just do random picks, and whenever that specific instance was not in my database, that was labeled as a negative example. I picked the number of yeah, I pick the same number of negative as instances as I have positive, just to avoid some class imbalances problems. As for my model architecture, I am creating a share embedding for my CDR3 and my epitope. So that's going to be my input data, which I am splitting into 80% training and 20% testing. And my model architecture consists of two layers, two convolutional layers, followed by one, fully connected layers that have 128 neurons, and then one final output neuron that's gonna do the prediction. Then I train my model, and after doing the evaluations, you can see that the model behaved really well, and the majority of the cases were predicted correctly, as you can see in this confusion matrix. Then if we take a look at the rock curve, you can see it's almost the perfect classifier. As you can see that we're able to achieve a really high true positive rate followed by a really low false positive rate, which is what we want. Then if we further look at the precision recall curve, yeah, again, we can see that we are able to get a really high precision and a really high recall as well. Then if we take a look at the distribution of predicted probabilities, here you can see all the cases where the actual label is no binding, and yeah, you can see the predicted probabilities are really below the current threshold, which is 0.5, and the opposite occurs for the instances that are positive. Then to obtain some biological knowledge of this, what I did was to pick, I give an example, a TCR, this is the TCR sequence, and the epitope. And what I did was to do, yeah, simulate mutations of all the positions of the CDR3 sequence. Then you can see here in green the actual position and here all the possible amino acids. And then after doing this mutation of this individual residue, I predicted the binding probability. And you can see that for some cases, you can increase the binding probability if you mutate that amino acid, or you can reduce the binding probability depending on the position. Some to the limitations of this study is that we're not considering the alpha chain. That is one important thing to keep into yeah, to keep in the top of your head, because the alpha chain is also part of the T-cell receptor, but yeah, the beta chain is the one that has been mainly characterized, but also important. Also, another limitation is that we're not considering the HLA, which is a plate that serves the antigen so that the T-cell receptor can recognize the antigen. We're not considering that. And then another potential limitation is the model. I'm using CNNs, but there are also some really good models that are able to do this task. Yeah, I think they would make a good job, such as RNNs and transformers. That's it. Thank you.